So I'm going to be talking about GenomeSpace, and in particular the crowdsourced analysis recipes and the recipe server that we've built to work with that. So genomic data comes in several varieties with data types such as DNA, RNA and protein sequence, expression data, interaction maps, and phenotypic data. Integrating these different types of data is difficult, but when you do it, you get a much more comprehensive picture of the underlying biology in an organism or system. However, it's not so easy to do. So colleagues of ours, Howard Cheng, created a stem cell paper in 2008 on the modular map of stem cells as they evolve into epithelial stem cells. And we created a graphical illustration of the workflow from that study over here on the right. Every one of the circles you see is a different tool. There are 6 different tools involved in this analysis. And every time you see a different color, that's a different tool. So every time there's an arrow between the tools, that basically reflects a case where somebody had to export the data, possibly reformat it, and then upload it into the new tool. This is a source of friction in the scientific process that slows down discovery. And this is one of the sort of things, the sort of problems that GenomeSpace is meant to address. So to address challenges in doing analyses like this, we developed GenomeSpace. So the goal was to make biological analyses available to biologists of any computational background. So you didn't need to be a programmer to do an analysis like you just saw. And what it functions as is essentially a lightweight connection layer between the tools. It handles authentication between the tools and transporting data between the tools and even reformatting data automatically for you as it passes from one tool to the next. GenomeSpace is web-based and includes Amazon Cloud Storage by default, but you can also hook up any other cloud-based storage provider that you want to it. And then once you've done so, you can pass your data to any of the GenomeSpace-enabled tools. In addition, tools retain their native look and feel. We don't change the tools. We don't require them to sort of modify or reprogram to conform to us. We let them just sort of seamlessly blend in so that if you're used to using IGV, for example, it behaves exactly the same way. About the only difference is you load off of a slightly different part of the file menu when you're trying to pull your data in. So some of the core features that we built into GenomeSpace are that the tools retain their native look and feel. So if you're already familiar with any of them, you'll still be familiar with how to use it. We have automatic data transfer and conversion between tools. So where formats are similar but not the same, you don't need to download it, convert it in Excel, add a new header file, or drop a column. That all happens automatically for you. We have direct import of data from repositories and URLs. So if you wanted to pull data from Geo or ArrayExpress, that's basically trivially easy, just point and click. And you can share data with your collaborators easily, either to other individuals, to the public at large, or you can create groups of users and share piecemeal with them as you choose. And then the focus of the rest of the talk is the recipe resource itself, which provides guides and workflows for completing common bioinformatics tasks. And of course, we also have a F1000 research channel, so you can publish the recipes that you create. So as of today, we've got about 20 tools and data sources in GenomeSpace. The data sources range from gene expression data sets to genomic coordinate data from UCSC. And the tools themselves provide a wide variety of genomic analyses. Some of the tools have hundreds of analyses just in and of themselves, like Galaxy and Gene Pattern. And we also include some of the most popular genomics tools that exist, such as Cytoscape, Gene Pattern, IGV, for example. Certainly we're going to have a few more tools as well, as we're talking and working with researchers at Penn to add Magic, which is for quantification of local splice variations from RNA-seq data, and with 7 Bridges to hook in their NCI Cancer Genomics Cloud Pilot into GenomeSpace as well. So there really isn't just one GenomeSpace anymore either. So for the most part, I'm going to be talking about the public GenomeSpace, which has been up and running for many years now. But if you live in the other side of the equator, GenomeSpace Australia exists. It's part of the Genomics Virtual Lab that the University of Melbourne runs. And the NASA GeneLab project is also spinning up another new GenomeSpace as well to be the analysis and data transfer backbone for that project. Now, when you go to genospace.org, that's sort of the starting point for anybody who wants to get into the system. You'll find that we have a bunch of helpful information there for people, so news highlights, blog posts, social media connections, etc. as well as documentation for tools and recipes and how you can use the system. So to start, you're going to click, To start, you're going to click on the big green user login button. And what that will do is bring up the GenomeSpace user interface. Now, this looks a lot like, the Windows Finder the Mac Finder or the Windows Explorer, but it's got some other abilities as well. So along the top, we also have a little toolbar here. This is where all of your tools exist. And you can customize this toolbar to put things on it, remove it. You can create additional toolbars if you want to organize tools by project. We have the area where you can manage your data in the various different cloud storage systems as well. And then if you want to actually launch, say, Site-Escape, you can just click on that icon and it will launch automatically, or you can drag a file from the file area up and drop it onto a tool icon, and then it'll launch that tool with the data that you've just provided. And lastly, if you want to upload files into GenomeSpace to put them up into your cloud storage, you can just drag and drop anywhere into this window, and that will start an upload automatically. So now moving on to recipes. There are a lot of tools there, and that doesn't necessarily make it easy to use. So what will make it easy to use is learning how to use the tools together and collaboratively. And for that, we have created GenomeSpace Recipes. Sure. Recipes are essentially, we might need to turn the gain down. Recipes are essentially the basic building blocks of analytical research. They're short, standalone workflows for performing common analysis tasks with just 2 or 3 tools typically. So with recipes, we basically modeled this on the classic lab manual resource molecular cloning, which describes short lab protocols that can be combined into larger experimental studies. Similarly, recipes can be strung together into more complex analyses. So our goal with the recipe resource is to create sort of an in silico version of molecular cloning in which we make common genomic analysis recipes available to the community to help drive research forward. And we find inspiration for these recipes from our own research, from published literature, and from the community. So when you enter the recipe resource, you'll see this page, and we have links to the normal sort of things you would expect. So there's a little navigation, there's a variety of documentation to help you use recipes or author them yourself. We typically have a featured recipe that changes periodically up front. And then we have the search area here, where if you were to say go in and search or say list all recipes, you'll start seeing the list of the available recipes for you to look at. Now from here you can look at just the titles of the recipes. If you click on the little greater than signs, that will expand that and give you about a 1-paragraph description of each of these recipes to give you a better idea of what it is before you dig in. Or you can cut down this list to filter it by either entering some search terms up here or using any of the filters. So you can look for things based on the type of input data they use or which tools they use or whatever tags that have been given to them, for example. Now, if you click through to a recipe, you start to see something like this. So recipes are made up of several modular components. So at the top, you'll have the basic sort of thing, like the title, a little cartoon image that gives you a rough idea of some of the behavior of the recipe, the author when it was last updated, for example, the tags that have been placed on this recipe, and then also a little thumbs up and thumbs down. Those represent upvotes and downvotes. So if you like a recipe, you can vote it up just by clicking on the thumbs up, or if you think it's not so good, you can do the opposite. And then, of course, there's the navigation area, and this gives you an idea of what most of the components of the recipe are like. This is an active navigation, so you can click on it to jump around within the recipe. And then, of course, you have a summary where you will basically describe or see a description of the motivation for the recipe, describing the biological problem that is being investigated in some detail. So as you scroll down this page, we continue with a description of the required inputs, where you can download example input data sets, which is also useful, and a description of the output data type and format. And then that's followed by a brief list style overview of the various steps in the recipe. Now, if we continue going down, we get to the actual recipe steps themselves. So in this particular recipe, it's a relatively simple one consisting of just 2 tools, Genomica and Site-Escape. And if you were to click on a step description, you'll see that the details of the step opens up. And this can include just expository text, it can include screenshots or images, or it can even include links to videos or video playlists typically hosted on YouTube, where you can actually watch the analysis being performed. Then after the final step of a recipe, we have a results interpretation section to review the implications and outputs of the analysis that we've just performed. Now, in some cases, there's more than one way to do a particular analysis step. And in this case, what you'll see is alternative steps. So at the bottom of a step, sometimes you'll see something like this, where there's another step, essentially an alternative step listed, identified by the different background color. Other than the background color, it's essentially got all the same information and multimedia that a normal step would have, but it's going to describe a different way you can do it. This might be because you're using different sorts of input data or different tools within GenomeSpace that can accomplish the same goal. And then finally, at the end of the recipe, you get the comment section where any user can go in and create comments on the recipe. Whenever you do create a comment, the author of the recipe and the GenomeSpace team are automatically notified by email, so we can go and take a look and respond to it as necessary. And one little error in this, one little error in this, the delete this comment, that's actually only available to administrators, and that's there just in case, sometimes people post nasty stuff or spam, so that's not normally used. So finally, if you wanted to create a recipe for yourself, how would you go about doing it? So to start, you need to log in. So you would use the same login as you use for GenomeSpace generally. It uses the same authentication system. Click on My Recipes, and you come to the page where you can see any recipes that you've already created, in this case none, and any that you might have voted up on or made a favorite out of. So not surprisingly, if you click on the big Add a Recipe button over here, that's going to be the entry point to essentially a simple form-based wizard that will help you create a recipe. So you'll see the information here is about the same as what you saw when you were looking at the recipe. Now you'll have your basic title. You can create that little title image, the one that shows up next to the recipe showing the tools, or if not, a default one will be provided. Optional video playlist, and then you can enter your summary information, for example, to describe it, just to describe the recipe briefly. Now, a recipe is not interesting, though, without steps, so you would then start to click the Add a Recipe Step button, and then you start getting additional forms popped up as well like this. So here you can, again, define your step title, optional video URL, and you can see that we assume that your recipe is going to probably be using one of the tools that's already in GenomeSpace or the GenomeSpace core features itself, which all show up in the pop-up here, or sorry, the list selection here. But if you want to use something else, maybe it's a tool that you've added privately to GenomeSpace for yourself only, or a tool that's outside of the environment, like you're making some steps in Microsoft Excel, you can annotate that here just as well, and it'll be added in as well. And then a brief description of the step, followed by the content section where you can go into as much detail as you feel is necessary, add links, screenshots, etc. Also, you can go in and add alternative steps. At the bottom of that step, you have an option to add alternative steps, so you can go in, create a different variation on that, whether it's based on different input data or a different tool with overlapping capabilities. Or maybe different parameter settings for a different analysis condition. Now, following this, when you're done, you get to the very bottom of the page and you're given a couple of choices. Now, you can save the recipe for further editing. If you do this, everything you've written in has been saved. You can go back at any time now and continue to edit it. So if you want to add steps, remove steps, reorder steps, alter the text, add images, etc., that you can do at any time. And when you're finally happy with what you've done, the idea would be that you would go and click the Submit for Review and Publication button. Now, when this happens, the GenomeSpace team, again, gets an email, and we're going to go and take just a quick review of the recipe. We are not reviewing it for scientific validity. We are making no judgments of whether we think it's good science or bad science. All we're doing is making sure that you're not some bot that has figured out how to submit our form and put spam in. And as long as it's not that, it will be approved probably within half an hour to an hour. And at that point, it'll be up on the recipe server for anybody in the public to view and comment on. And that is actually how we do sort of manage the scientific validity of experiments, is rather than us judging it, we just count on people adding their own comments and letting them discuss the recipe at the end themselves. Now, once you've published your recipe, the final step is we would like to encourage you to publish it again on the F1000 research channel. This gives you a citable reference to the work that you've done and also makes it a lot more easy for other researchers to find it and benefit from the work that you've done. And then as an aside, from our group, we have a new recipe that's going to be released very soon, hopefully by the end of the summer, and its title will be Identify Essential Single Guide RNAs and Genes from Genome-Scale CRISPR-Cas9 Knockout Screens with Magic. So hopefully that'll be out by the end of August, and that will be the next one that'll be released from our group. But again, anybody in the public can go in and create recipes as well. And with that, I think I have a couple of minutes left for questions if anybody has any.
So thank you very much, Ted. Are there any questions, comments? Good. There's a mic there if you want to. Oh, yeah.
Thank you. Do you have any sort of testing frameworks to make sure that the recipes work, or when the data behind them changes, that people might be notified of that?
So no, our testing framework really comes down to a couple of junior researchers in our lab who we detail to replicate things. Actually, to be honest, one of the ways that gets done is whenever a new person joins our lab, one of their first tasks is always go run through a couple of recipes, and we'll sort of assign the ones that we think might have varied a bit.
Okay, thanks Ted, thanks Ian for that question. I've got one more question. If you'd like to go up to the mic, that's great.
Maybe you can comment on the type of files that are typically uploaded and about the size of the files that can be processed by the GenomeSpace.
Okay, so GenomeSpace doesn't do the processing itself. All the actual analysis are done by whatever tool you're working with, right? So if you're working with IGV, you can use BAM files as big as you can generate. And actually, IGV is a great example because it only does sort of random access into like BAM files. So even if you have like a 100 gig BAM file, it's only going to pull little bits of data out of it at a time and never actually download the whole thing. If you're using, Gene Pattern or Galaxy, both of those systems are pretty well configured to work with very large files. If you're using desktop tools like Genomica or GE Workbench, then they're probably a little bit more limited in terms of the size of the data they can use. Now the data itself is it's all hosted on the cloud. We provide a 30 gigabyte share of data to everybody when they join automatically. But you can also hook in anything else that you like. So for example, at UCSD and probably a lot of other universities, we get unlimited Google Drive storage. So we can hook our Google Drive accounts in, and now I've got unlimited storage. It still goes through the GenomeSpace authentication, so I can pass it to Gene Pattern or Galaxy or whatever without re-logging in. But yeah, I have essentially no limit on what I can put in.
Okay, so thanks very much, Ted. Thanks so much for those questions. Thanks.